Hello, everyone, and welcome to the Connect Science webinar. I'm David Weinstock. I'm very happy to welcome Dr. Frank Slack, who will speak to us today on personalizing microRNA therapeutics in cancer. Dr. Slack is a preeminent scientist in this field. He's a professor of pathology at the Beth Israel Deaconess Medical Center and Harvard Medical School. And he leads the Harvard Medical School Initiative for RNA Medicine. A couple of brief announcements. First, I apologize for the last minute addition of a password. We were just notified that we needed to have a password because of the larger issues around people Zoom bombing. So as unfortunate as that is, we're going to post the password on the website, and we will get the information around the password out as widely as possible. Please do feel free to email anyone you know who's interested in these lectures and have the password passed around as well. We're very fortunate tomorrow to have Dr. Ross Levine from Memorial Sloan Kettering Cancer Center, who will be speaking on clonal evolution. And speaking of Memorial Sloan Kettering, they are also hosting an internal webinar called MSK Science Spotlight with preeminent investigators giving lectures. So I encourage you to take a look online as well. Again, that's called MSK Science Spotlight. As always, we thank the Leukemia Lymphoma Society and the Dana Farber Harvard Cancer Center for helping to support this webinar, which as of yesterday had over 20,000 total attendees across the lectures and downloads thus far, which I think anyone would consider to be an enormous success. Please post any questions using the Q&A tab at the bottom, and Dr. Slack will be happy to address them. And at that point, I'll stop talking, Frank, and let you take it away.
Great. Well, thank you to David and Nellie for the opportunity to speak at this great seminar series. I'm going to start just by giving a shout out and a big thank you to all of the healthcare workers at the BI, as well as to all of the healthcare workers at the hospitals throughout the Massachusetts area, and in fact, throughout the world, for saving lives and putting their own lives at risk at the same time. So we thank you very much for your work in this difficult time. So today I'm going to tell you about my lab's efforts and the field's efforts towards moving discoveries in non-coding RNAs into personalized medicines. And in particular, I'm going to focus on one class of non-coding RNAs called microRNAs and their role as potential personalized therapeutics. I hope to do this by providing you with a few vignettes from my own lab. But as many of you know, I'm a developmental biologist by training, and so I've had a lot of help from collaborating labs, some of which are shown here in this slide, including a number of biomedical engineering labs, organic chemistry labs, and clinical collaborators who have not only helped us be able to deliver some of these agents, some of these novel agents to cancer tissues, but also to provide us with information on unmet needs that maybe these new types of therapeutics might meet. And then by way of disclosures, I'm a founder or a consultant for many of the companies that are working in this space, or at least for some of the companies working in this space. So of course, there's a lot of excitement these days around non-coding RNAs. They've been recognized for about 15 years now as being a new paradigm for gene regulation. And then over the last decade or so, they've been recognized as a new paradigm for diagnostics and therapeutics. And my lab initially focused very heavily on this part of the equation, trying to understand their role in gene regulation. But for the last few years, I'd say the last maybe 10 years or so, we've really been trying to get us from here to here. So how can we harness these new discoveries for new diagnostics and therapeutics? As I said today, we'll focus mainly on the therapeutic angle, although there is a lot of work obviously going on in the diagnostic space as well. So RNA medicine involves non-coding RNAs. Non-coding RNAs are one part of RNA medicine. RNA medicine is undergoing a renaissance right now. You've probably heard about some of the amazing successes from 3 of the Boston area pharma and biotech companies, including the first RNAi drugs from Alnylam, the first, or at least an antisense drug from Ionis and Biogen called Spinraza that can treat spinal muscular atrophy. It's an RNA targeting oligonucleotide. And then most recently, the work from Moderna, which includes an RNA-based vaccine against the SARS-CoV-2 virus. It's my hope that these early studies have sort of opened the floodgate for novel RNA-based medicines, of which non-coding RNAs should play an important role, and I'll talk about that as we go through the talk today. Another point I want to make is that RNA medicine is getting highly personal. Because RNA medicine essentially involves base pairing between an oligonucleotide and an RNA, or between RNA and DNA, its ability to be personalized is very apparent. And this is illustrated very nicely by this report from Tim Yu's lab at Boston Children's Hospital, where they designed an oligonucleotide that would target a mutant RNA that, as far as I knew, was only found in a single patient. Her name was Mila. And they termed this drug Milasin based on the fact that it was used to treat this patient. And as reported in the New England Journal a few months ago, this particular oligonucleotide therapeutic is stabilizing this child's disease. So you can imagine now that many, many more personalized RNA medicines are going to be making it to the clinic in the next few years. Now, the Human Genome Project and the ENCODE project showed us that we have a large proportion of our genome that doesn't code for proteins. In fact, we have about the same number of protein coding genes as do simple organisms like C. elegans and fruit flies, and yet we have obviously a lot more complexity. But we have a genome that's about 30-fold larger than those organisms, and much of that genome space is made up of non-coding material. The ENCODE project in around 2012 showed us that, in fact, much of that non-coding portion of our genome is, in fact, transcribed into RNA. And we're trying to make sense now of what those non-coding RNAs might actually do within cells. Now, since that early work, we now know that the human genome produces a number of non-coding RNA classes, including the microRNAs, of which I'll talk to you about today. And there are at least 2,500 individual species of microRNAs that are encoded within our genome. Now, this is made more complex by the fact that many of these microRNAs are modified either post-transcriptionally or at the genome level into isomeres, and these isomeres constitute about 250,000 different varieties of microRNAs within our cells. Within our germline, we produce about 100,000 Kiwi-associated RNAs, or piRNAs, and these are very important for silencing transposons and for genome integrity in our germline. Given the fact that we now know that there are 48,000 or so long non-coding RNAs that are produced from our genome, we can now say that we have almost 2 times more long non-coding RNAs than we do protein-coding RNAs within our cells. Those 20,000 pseudogenes that we all learned about in high school and college biology as being sort of inert relics of an ancient gene, we now know many of those are transcribed and can have functions. And more recently, the discovery that there are about 100,000 circular RNAs and 25,000 tRNAs found within cells has really added to this amazing complexity that we now see within the non-coding space in our genome. Now, this complexity is going to provide a lot of material for scientists to work on over the next few years. Of course, we want to understand what all of these RNAs are doing. The way I see it is that this complexity turns into an amazing amount of opportunity. We can now attack disease. We can now use these as biomarkers for disease in ways that we have a new arsenal of genes that we might be able to use in disease remediation. So I see this complexity as an opportunity. So what are the opportunities for novel therapeutics? So the way that I've viewed this, or at least the way my lab has viewed it, is we see that 3 different opportunities, 3 different immediate opportunities. The first is that the RNA can be a drug. And so you can imagine an example where maybe an RNA is lost in a particular cancer patient, maybe the gene is deleted or the gene is epigenetically silenced, you might consider restoring that particular RNA, for example, if it was a tumor suppressor RNA. In a second example, the RNA can be drugged. So one could imagine that if the RNA was amplified or had some oncogenic role in a tumor, you might be able to inhibit that RNA. And we think of multiple ways of inhibiting these RNAs, but the most common one right now is to use an antisense type approach. But there are now colleagues in the field who are using small molecule approaches to try and target oncogenic RNAs as well. And then in the last case, which I won't talk about much today, there are ways to inhibit the biogenesis of these offending RNAs through small molecule drugs. So given that non-coding RNAs are so prevalent in human tissues, and because many of them are tissue specific, and I won't have time to talk about that much today, there are a growing number of clinical trials that are happening involving non-coding RNAs. I recently just did a search for one class of non-coding RNAs, the microRNAs, in clinicaltrials.gov, and I found that there are about 900 clinical trials involving microRNAs at the moment. So as shown by these first four, many of them are in the diagnostic space, but there are probably 24 or so in the therapeutic space as well. And I'll mention some of those as I go along today. So I'll talk about microRNAs, and particularly their roles in cancer, but many of the concepts that I talk about today are likely to be applicable for other classes of non-coding RNAs and for other diseases as well. In fact, other people have proven that to be the case. So let's just talk a little bit about the background for microRNAs in cancer. They were shown by Carlo Croce and George Kalin many years ago to be frequently located in genomic regions that were linked to cancer. So in regions of loss of heterozygosity or amplification or deletion or regions of epigenetic modification. They're frequently misexpressed in tumors relative to normal adjacent tissue. And this has allowed them to become very useful biomarkers for cancer diagnosis and prognosis. We now know that there are mutations in the members of the biogenesis machinery that are involved in microRNA biogenesis, including very well-known enzymes such as DROSHA and DISA. Those enzymes are frequently mutated in cancer. We know that microRNAs fit within cancer pathways. They regulate oncogenes, and they're regulated by well-known cancer factors such as transcription factors like MYC and P53. And in fact, they themselves can act or can be tumor suppressors and oncogenes. And I'll give you some examples from our own work where we've built mouse models and shown that mouse models missing or overexpressing some of these microRNAs, in fact, get tumors. So the advent of large-scale whole genome sequencing approaches has shown that there are mutations within microRNA genes, as well as within the microRNA binding sites in their targets that are found in tumor patients, found in tumors from cancer patients. And this whole area has sort of become the sort of new battleground in trying to assess whether there are mutations in non-coding portions of the genome that can be informative for cancer diagnosis and cancer prognosis. Now, lastly, microRNAs are stable in many bodily fluids, such as urine, serum, breast milk, etc. And they're acting as diagnostic and prognostic biomarkers. And in fact, there are a number of clinical trials involving measuring the levels of microRNAs in bodily fluids as diagnostic and prognostic biomarkers. So the first vignette that I want to talk about today is this idea of using microRNAs as therapeutic targets. And the example I want to use today is a microRNA called miR-155. miR-155 was shown by a number of labs, including our own, to be highly expressed in cancers of sort of liquid cancers like lymphomas and leukemias. This microRNA miR-155 is known to be involved in the genesis of the hematopoietic system. So in order to try and show that miR-155 was in fact an oncogene, we built a mouse model where we could induce the levels of miR-155 through the addition of doxycycline the hematopoietic system. And when we did that, we found that the mice develop leukemias and lymphomas. The lymphoma is very well, sorry about that, go back. Lymphoma, of course, is possible to see here in this mouse as this large outgrowth that's growing out of the side of its neck. This is an axial lymph node that's, that has lymphadenopathy. So this proved to us, or proved to me, that miR-155 could in fact be an oncogene. One of the remarkable features of this particular mouse was that we now had a tool to test strategies for knocking down or knocking out the activity of miR-155 and asking whether this could be a viable target for leukemias and lymphomas. And when we did that in two different ways, either by adding, by changing the levels of doxycycline in the food so that the microRNA was now no longer expressed, or by targeting it with an antisense molecule that was antisense to miR-155. We found that, in fact, within just a few days, these tumors completely regressed and these mice were completely healthy. So this proved to us that not only was miR-155 an oncogene, but that these tumors depended on the continual expression of miR-155, that they were addicted to miR-155, and in fact, miR-155 could therefore be considered a viable target for an anti-cancer therapy in lymphoma and leukemia. And this work led to a clinical trial from a small biotech called Murigen out in Boulder, Colorado. This clinical trial involves an antisense to miR-155 called MRG-106. It's also called cobamarzin. This trial opened initially in patients with T-cell lymphomas, including cutaneous T-cell lymphomas, as well as the T-cell lymphomas caused by HTLV-1. And I won't have time to go through the results of those particular studies. Safe to say that this particular drug is now in phase 2 trials for those types of lymphoma, and has recently opened in other types of bloodborne cancers, such as diffuse large B-cell lymphoma and AML. So just to get back into the science for a minute, we had a few interesting questions that we wanted to ask using our mouse model. The first was, how does miR-155 cause cancer? And the second is, why do these cancers become addicted to miR-155? So using gene expression profiling, we had some sense that the mechanism of oncogenesis was likely to be because of the individual targets that miR-155 downregulated. We knew that miR-155 downregulated some signal transduction molecules, including SHP1 and SOX1. And in addition, it downregulated genes involved in DNA repair and in the mutated phenotype, including BAC1, TP53, INP, and the mismatch repair genes and DNA polymerase delta. So in mice or in human tumors that have overexpressed miR-155, you get a decrease in this negative signal transduction pathway, as well as an increase in these genes leading to a mutated phenotype. However, our model suggested that there would be a two-hit mechanism for this type of leukemia and lymphoma that was manifest in our mouse. First of all, we noticed that the tumors were clonal. So in each individual mouse that obtained a tumor, we were able to show that it appeared that a single B cell had was responsible for the tumors that and all the metastases that were found within that particular mouse. And secondly, we were able to show that there was a variable time to onset. Each mouse became sick at a slightly different time, such that at about 75 days after miR-155 was turned on in all of the animals, about half the animals had shown tumor formation. So this suggested to us that there was a two-hit mechanism. So in order to try and determine if there was a second hit that was occurring in these tumors, we did whole genome sequencing on, whole exome sequencing on different tumors from different mice. We found one common gene that was mutated in almost all of these tumors, and that was the gene KIT, the oncogene KIT. KIT, of course, is a receptor tyrosine kinase. All of the mutations lay in areas of the tyrosine kinase that would predict it to be activating mutations. And in fact, we were able to show that in 4 different tumors here. They all activated signaling through the KIT pathway by sort of phospho-STAT3. Sorry, that's my doorbell going. If you can hear something in the background, I apologize. So we had this opportunity now to ask whether these tumors were in fact also dependent on KIT signaling, and in fact we found that they were. We could see radical tumor regression when we treated the animals with a KIT inhibitor, dasatinib, or a different inhibitor called imatinib. And you can see the reduction in the tumors here. Those large lymphomas by day 9 are pretty much gone in these animals after KIT inhibition. This is similar to what we saw with miR-155 inhibition. So we ended up with this very interesting situation now where it appeared that these tumors were addicted to two independent, or at least apparently two independent oncogenes, miR-155 and KIT. It was strange to us to think that even though these tumors had activating mutations in KIT, they were still dependent on expression of the oncogene miR-155. But this conundrum was actually solved when we looked at whole RNA-seq from these animals. What we found was, interestingly enough, KIT was the most differentially expressed gene in tumors that were forming versus tumors that were regressing from these mice. And in fact, it appears then, and you can see from this Western blot that in the absence of miR-155, so when we add doxycycline, we shut off miR-155, KIT expression completely disappears in these tumors. So it appears that not only is KIT potentially involved in the mutator phenotype that allows for mutations to occur in KIT, but miR-155 is also involved in the expression of KIT. And we used a bioinformatics approach to ask which potential targets of miR-155 might be involved in that. And there are two transcription factors well known to be involved in leukemias and lymphomas, SPI1 and MIB, which are known to upregulate the transcription of KIT. And miR-155 is a negative regulator of both of those factors. And those factors are also changed in our gene expression analysis shown down here with the tumors that are forming versus regressing. So this is evidence for a multi-hit mechanism of oncogenesis by miR-155. It's kind of similar to some of the other reports that are coming out that in certain lymphomas and leukemias, it requires multiple hits. In this case, we think that an oncogene is cooperating with oncogenic mutations in another well-known oncogene called KIT. We think that this is not just cooperation, but in fact, miR-155 promotes KIT activity because it also regulates the expression of KIT, and there's sort of a feedback loop between these genes. There are cases in human oncology where miR-155 and KIT expression are both high. For example, AMLs have high levels of miR-155 and KIT, and so we posit that this mechanism might be applicable for acute myelogenous leukemias. Of course, I've just mentioned that MIB and SPI1 are the most likely targets for miR-155 in this pathway. We posit that this type of model could apply to other microRNAs that promote a mutated phenotype. And another one that comes to mind is miR-21, which I'll tell you a little bit more about later in this seminar. But the overarching conclusion for this part of the work is that oncomers can be viable targets in cancer, in particular in cancers that have overexpressed those particular oncomers. And what I failed to mention is that the Miragen trials are biomarker-based, so the patients do get their miR-155 levels tested before they go on trial. So the second example that I want to talk about today is the sort of flip side. Can we use microRNAs as targeted therapeutics? So in this case, potentially delivering a microRNA that's a tumor suppressor to a cancer patient that might have lost that particular microRNA. And the example that I want to talk to you about today is the example of miR-34. It was actually first identified in the C. elegans model, but was quickly shown to be highly conserved in sequence throughout phylogeny. And in fact, you can see the human miR-34 and the C. elegans miR-34 are extremely well conserved. And in fact, it was shown that miR-34 is a direct downstream target of p53 and very important for the DNA damage response. So work from our lab and other labs has shown that miR-34 regulates at least 40 different oncogenes and immune checkpoint genes. I won't have time to talk much about the background to miR-34, to say that's a very promising target for microRNA replacement in a number of different tumors, in particular tumors that have lost or have mutated P53. So some of the initial enthusiasm for this work came from an experiment we did a few years ago, where we used Tyler Jacks's KRAS P53 double mutant lung cancer model, where you can see that when you induce activating mutations of KRAS or dominant negative mutations in P53 into these mice, the lungs show very rapid development of tumors. If you dissect the lungs, you can see multiple lung nodules forming in these mice. So without going into all of the background to this, this one experiment I think illustrates some of the power of using miR-34. When we introduced miR-34 into this particular mouse model and activated these two lung cancer genes, we found that we could completely suppress the formation of lung tumors in those mice, suggesting that miR-34 can bypass the loss of P53 in this mouse model. And so in continuation of this, we were able to show that systemic delivery of miR-34 in a lipid nanoparticle had a similar effect. And that led to a clinical trial with a company called MICRORNA, using a miR-34 mimic delivered in a liposomal formulation. This particular trial, as shown here in red, was terminated early, or at least I should say it was terminated at the end of the study, when at the end of the study, a few patients developed serious immune-related adverse events. While the results of this particular study have not been published, I have been told that some of these patients developed cytokine storm, and so this particular study was terminated. So I don't feel like this particular target has been invalidated. I just feel like there are approaches that we can take to make this particular therapeutic modality more effective. And so what are the things that we can do to make this safer and more effective? And that's what I'll tell you about in the next few slides. In the first case, we'd love to improve the delivery. Instead of delivering this systemically, as was done in this trial, and of course, delivering mostly to the liver, we would like to be able to improve the delivery, either in vivo delivery to the tumors themselves, or ex vivo delivery, where we can maybe potentially work with cancer cells or with immune cells that have been removed from the patient and then put back into the patient. I should say that these immune-related adverse effects were seen at high doses of the MRX-34. And so one strategy that we need to try to develop is ways to lower the dose. And so we need better chemistry to improve this efficacy, to improve the pharmacokinetics of the drug in humans, as well as to reduce the immunogenicity of these double-stranded RNA drugs. And then lastly, we've looked for rational combinations for drug synergy. So we can use lower doses of miR-34 in combination with some maybe standard of care synergies. I'll talk about each of these three in the next few slides. So just to say that we have partnered with oligonucleotide chemists, like for example, a wonderful chemist at UMass Medical School, Anastasia Krovera, to try to modify these microRNA mimics to make them both more effective and to reduce their immunogenicity. I won't talk about what these modifications are, but they are sort of standard type third generation modifications that are now found in some of the siRNA drugs that are in patients right now. And so these same types of modifications we've now shown can be introduced into microRNA mimics and can be effective in vivo. So with the ICCB at Longwood, we screened for drugs that could synergize with miR-34. We screened through about 10,000 different compounds, some of which are FDA approved, for drugs that could both decrease lung cancer cell viability or increase apoptosis of these lung cancer cells only in the presence of additional miR-34. The top 50 of those hits are shown here in the middle. You can see that for the most part, these reduce the viability of the cells, and in many of them also increase apoptosis in those cells. We took these top 50 and we performed dose studies with them, as well as looking for combination index type studies. And one of the drugs that I'll talk to you about today that emerged as the strongest combinatorial partner with miR-34 was this drug, Wabane. Wabane, you can see by these CI index plots, synergizes with miR-34 here in two different cell lines, A549 and HCC 827. In fact, in multiple different cell lines, we could shift the IC50 of the microRNA using Wabane as a synergizer. Now, Wabane is a drug that is currently FDA approved for certain cardiovascular indications. We do not yet know exactly how Wabane is working to synergize with miR-34, but I can tell you without going into details, the details are in this paper, that the combination of these two factors induces massive autophagy in the cells and causes them to have decreased cell viability. So we're currently working on Wabane and some of the other compounds that came out of this study to see whether these might be useful to add in combination with miR-34 in future clinical trials. So in terms of trying to use microRNAs as a potentially ex vivo, we've asked whether we could use miR-34 as an immune checkpoint inhibitor. And of course, immune checkpoints are extremely hot therapeutics right now, but there are still barriers to successful immune checkpoint inhibition in oncology. At this point, only about a quarter of patients that receive immune checkpoint inhibitors will show a positive response. And in addition, many patients are going to develop autoimmune toxicities against these drugs. So we and others have shown that, in fact, microRNAs regulate many of these immune checkpoint genes. In fact, miR-34, we and others have shown, directly represses the expression of the PD-L1 immune checkpoint gene. PD-L1 shown over here, which is the binding partner for PD-1. As shown in this paper over here from our lab, we were able to show that, in fact, diffuse large B-cell lymphoma cells that were treated with a miR-34 mimic showed reduced expression of PD-L1. The quantification is shown over here. Without going into too much detail, on the next slide, we teamed up with Chris Chen at the BUV's tissue microfabrication lab to design a sort of a lab on a chip experiment where we try to mimic a tumor microenvironment for cancer cells and activated T cells. And what we found here in this particular setup was that the cancer cells, which are shown here in green, and the activated T cells, the CD8, CD4 cells shown here in purple, do interact with one another, and there's sort of a T cell killing that occurs at a low level without any alteration in miR-34 levels. But when the cancer cells were pre-treated with miR-34, the same contraption, what we found was that there was increased T cell killing. There was increased recruitment of T cells, increased T cell killing, and of course, reduction in the number of cancer cells that were found in this particular system. So this was, for us, sort of a proof of concept that miR-34 might potentially be used ex vivo in a manner that allows it to be an immune checkpoint inhibitor. And we'd love to be able to move forward with those kinds of studies in the future. So given the increased interest in non-coding RNAs in cancer and all of those new discoveries of hundreds and thousands of new non-coding RNAs, about 6 years ago now, Harvard Medical School began this HMS Initiative for RNA Medicine. Our mission is to make and translate RNA discoveries into novel therapeutics and diagnostics by fostering a climate of outstanding basic translational and clinical research with world-leading scientists and clinicians, both at Harvard Medical School, but also in the Boston area with some of the. So the HIRM currently has a number of initiatives. I would say that these are, generally speaking, a three-pronged approach towards combating different diseases. I'm going to tell you about our efforts to combat cancer, but they involve, of course, discovery, moving into the clinic, and overcoming delivery issues for the particular tissues that need to be targeted for each individual disease. So for the discovery effort, we need a massive discovery and validation effort towards revealing and understanding the 98% of the genome in disease, both for novel biomarkers and for novel therapeutic targets. We want to integrate this non-coding RNA data into the patient standard of care. We call this the 98+2 initiative, for not just trying to target the 2% of the genome that's protein coding, but also taking into account the other 98%. We'd love to offer RNA diagnostics and the possibility of custom novel RNA-based therapeutics that target the uniqueness of each and every patient. And I'm going to spend the rest of my talk telling about how we're getting to that in at least two different examples today. Of course, we need to overcome RNA delivery barriers, given the fact that these RNA therapeutics are typically large molecules, 22 nucleotides in length, typically, with high charge values. And so we need to learn how to overcome these delivery issues. I should say that with the help of biomedical engineers, we are in fact solving some of these issues. You can see an example here of a mouse in which we've targeted an oligonucleotide just to lymphomas in this particular mouse. So in order to help us, as well as additional people that want to join us in this effort, we've started a non-coding RNA precision diagnostics and therapeutics core at BIDMC. We think of this as sort of offering a full non-coding RNA concierge service, from detection to function to therapy. And to do that, we have 3 units currently in operation in the core. By the way, the core is open to everybody in the community, so please feel free to reach out to us. We have a detection core, which specific detection of non-coding RNAs within clinical specimens or any kind of specimen that scientists are interested in studying. We have a bioinformatics unit, which provides specialized bioinformatics for interpreting non-coding RNA discoveries. And these include mutational analysis in the non-coding portions of the genome, or RNA-seq analysis, which specifically examines the non-coding RNAs in that particular data set. And then we have a delivery core, which allows our clients or our researchers to design novel nanoparticles that might allow them to deliver their RNA or their antisense molecules to the tissues that they're interested in doing. So we have a wonderful team shown down here, and we have technologies from these companies in the core at the moment, so please feel free to reach out to us. So to give you a sense of just how we're taking this idea and trying to move it into the clinic, I'm going to give you 2 examples from my group in collaboration with some of the clinicians that I mentioned earlier. The first is in pancreatic cancer, and the second is in non-small cell lung cancer. So the idea here is to perform or obtain a complete catalog of all the RNA transcripts that are found within normal and cancer cells from particular patient samples. And in the examples that I'll tell you about today, we've either used cancer cell lines from patients, or we've used patient-derived organoids or patient-derived xenografts. So we're trying as much as possible to use patient samples for these kinds of studies. We want to obtain a complete DNA sequence from each patient so that we can understand the non-coding portions of the genome. We will use the bioinformatics unit to stratify which targets might be actionable based on differential expression or differential mutation analysis between the normal and the cancer samples from each patient, and then try to functionally validate which of those targets might be actionable using patient-derived avatars, either PDOs or PDXs, or in some cases, if we need to, genetically engineered mouse models. So for the example of pancreatic cancer, I'll start with a study that we performed with Manny Hidalgo in profiling pancreatic cancer patients for their microRNA complement. Shown here on the left is the levels of miR-21 expression that we obtained from about 30 different pancreatic cancer patients that were in Manny Hidalgo's collection. What we noticed, interesting enough, was that some of these patients, shown here on the bottom of this graph, had normal levels of miR-21 expression, equivalent to what's seen in a normal pancreas. but that some of the patients, about 2/3 of them, showed increased levels of miR-21. So this led us to start thinking that maybe there was an opportunity here to target miR-21 in these patients that had high levels of miR-21. I'll give you a few reasons why we think targeting miR-21 might be fruitful in a minute. The first piece of evidence for that is that from the TCGA data in pancreatic cancer, you can see that patients with high levels of miR-21 have a poorer prognosis than those with low levels. I mean, even though pancreatic cancer has an extremely poor prognosis to begin with, having high levels of miR-21 makes your life even shorter. We already knew from mouse model work that miR-21 was an oncogene, and that mice become addicted to this particular oncogene. So similar to the experiments that I showed you before with miR-155, when we overexpressed miR-21 in the hematopoietic system, we saw lymphomas occurring in these animals as well. I should tell you that these are different lymphomas than what we get in the miR-155 mice, and in fact, they don't involve overexpression of miR-155 at all. So in order to try and target miR-21 in pancreatic tumors, we started a collaboration with Sangeeta Bhatia's lab over at MIT. Of course, pancreatic cancers are notorious for having a high stromal content and having very poor drug delivery. But we worked with her on a nanoparticle formulation that she'd already shown quite efficiently targets siRNAs to difficult to treat tumor sites like ovarian cancers and pancreatic cancers. And when we added an antisense molecule to miR-21 into this particular nanoparticle, which we called the tumor TPN for tumor penetrating nanoparticle, we were first able to show that in fact we could deliver the drugs into organoids that we were growing from these pancreatic cancer patients in the lab. So you can see here by the red dye that we were able to deliver the antisense into the center of these organoids, shown here in this, also in this confocal image. In these organoids, which look like this under the light microscope, when we delivered this, the TPN containing the antisense to miR-21, we were able to show that these organoids quickly dissolved or disintegrated. They underwent massive apoptosis, as shown here by this apoptosis dye. And in fact, using an in vivo model, we were able to show that in fact we could deliver anti-miR-21 using this nanoparticle into pancreatic tumors at about the same levels as we were seeing it go into some of the tissues that naturally take up some of these molecules, like the spleen. So this was an encouraging piece of evidence, and we went on to show that in fact mice containing pancreatic cancer tumors that were delivered these particular nanoparticles showed slow growth of their tumors. I'm not going to show you that evidence today. I'm going to move to the more personalized aspect of this work. So what we were able to do now is to ask, given that we had 30 different pancreatic tumor samples, avatar samples grown in the lab, could we differentiate, or could we show a differential response to anti-miR-21 in the tumors that had high levels of miR-21 versus the ones that had just normal levels of miR-21? As shown here, PDOs 286 and 219 have high levels of miR-21, whereas PDO 017 and 030 had normal levels of miR-21. Without going into too much detail, because the results are shown in this paper, we were able to show that only the tumors that had high levels of miR-21 would respond to an anti-miR-21 therapy. So in this case, PDO 286, which is in fact gemcitabine resistant, could respond to an anti-miR-21, whereas PDO 017, which is gemcitabine sensitive, but which has low levels of miR-21, didn't respond at all to the anti-miR-21 therapy. Now, I know this sounds particularly trivial, but it was the first example that we had of using this as an RNA-based opportunity for personalized medicine. So in this case, we think that the non-coding RNA is both the companion diagnostic and the therapeutic target. So sort of a theranostic, if you will. And this is the approach that Mirogen has taken in their clinical trials. So patients that are put on the anti-miR-155 trials are first monitored for the levels of the microRNA in their tumors. So what we noticed was that even though about 2/3 of the pancreatic cancer patients overexpressed miR-21 and a number of other microRNAs. There were some microRNAs that were only, like miR-196 shown here, that were only overexpressed in some of the patients. Let's say, for example, in this particular patient. And we're currently now testing whether there are additional microRNAs that could be potential therapeutic targets in these other cancer patients. And this, I think, is the sort of leading us to this promise of, of personalized microRNA therapeutics. And we'll be able to determine, based on gene expression profiles, which microRNAs might be targetable in a particular patient, and then using an avatar like a PDO, test whether targeting that microRNA might be a useful strategy in, or at least a useful therapeutic opportunity in treating those patients. So in the last example that I'll talk about today involves EGFR mutant lung cancer. So EGFR mutant lung cancer is a very interesting disease. It's normally found in non-smokers and involves mutations in the tyrosine kinase receptor EGFR. Now, patients with mutations in EGFR often respond to a tyrosine kinase inhibitor like erlotinib or gefitinib. However, these first-generation tyrosine kinase inhibitors often result in, often show a clinical benefit for a few months to a year, but the patients ultimately relapse with acquisition of second site mutations, sometimes within the EGFR receptor itself. A commonly identified second site mutation is this one, T790M, that occurs within EGFR that makes the EGFR now insensitive to the binding of these two TKIs. A third generation TKI called osimertinib was developed that was effective against mutant EGFR T790M mutant cancers, and in fact is now frontline treatment for EGFR mutant lung cancers. However, resistance to this third generation TKI is now emerging in the clinic, in particular with third site mutations now in the EGFR receptor gene at the C797S site. There are other mechanisms to bypass signaling through EGFR, even though it's been inhibited with these TKIs. There are additional protein tyrosine kinases that are activated. And lastly, hypoxia has been associated with drug resistance. In fact, part of that is shown here on the right. You can see that in a patient illustrated in A and B that failed EGFR TKI treatment, the tumor shown here in their CT scan, in fact, progressed on this particular tumor, on this particular regimen. If you look at a PET labeling using FMISO, which labels hypoxia, you can see that this particular patient has a highly hypoxic region around the tumor. In contrast, this patient here, who by CT scan responded to an EGFR TKI, you can see that the tumor actually regressed. Their tumor showed reduced hypoxia in the same type of analysis. And in fact, it has been shown by a number of scientists shown here on the bottom that upon EGFR TKI administration, there is a small subset of cells that seem to persist that are tolerant of the drug, and upon removal of the drug or upon second site mutations can now grow out into additional tumors. It's a very new field, and very little is known about targeting drug tolerance, but you can imagine that if you could prevent these cells from becoming tolerant to the drug to begin with, you might see better efficacy in this tumor type. So we asked whether there were microRNAs that might be involved in drug tolerance in this particular case. So we identified a number of EGFR mutant cell lines, including PC9 cells and HC827 lines, and we grew them in low levels of osimertinib. We also did this in the presence of gefitinib and erlotinib, but I'm just going to show you the osimertinib results today. And we obtained osimertinib tolerant cell lines of these two EGFR mutant cell lines. And then we profiled the levels of microRNAs from those particular cell lines, as well as we did RNA-seq analysis from these cell lines. One microRNA called miR-147B had previously not been shown to be involved in any form of drug tolerance or drug resistance, and so we focused mainly on miR-147B. Another reason for focusing on that was that when we looked at the TCGA data from EGFR mutant versus KRAS mutant lung cancers, EGFR was one of, miR-147B was one of the best distinguishing microRNAs for the EGFR mutant lung cancers over the KRAS mutant lung cancers. So in the parental cells, we had low levels of miR-147B. When we made them tolerant, the levels of miR-147B went up. We could take these cells off drug and they would regrow again. They could recover, and we found that the levels of miR-147B were low again. And then when we made them tolerant a second time, miR-147B levels increased. So this told us that miR-147B might be an important, at least a biomarker for tolerance, but could in fact be targetable in drug tolerance. I told you that we did RNA-seq from these same cells, and these cells express a pseudohypoxia signature. I'm not going to have time to tell you all about the RNA-seq analysis, or in fact what we did with the pseudohypoxia response, except to say that one of the main targets of miR-147B that we showed in this paper was the von Hippel-Lindau tumor suppressor gene, VHL. And in fact, lung cancer patients with high levels of miR-147B have a poor prognosis, as do lung cancer patients with high levels, in this case, of, with low levels of VHL have the poorer prognosis. So there was an inverse relationship between miR-147B and VHL in the clinical data, as well as in the gene expression data, which I won't have time to show you today. So if this was important in drug tolerance, then if we knocked out or if we overexpressed miR-147B, could we change the tolerance of those cells? The first experiment shown here is just if we overexpress miR-147B in cells that are normally sensitive, and these are lung cells that are normally sensitive to osimertinib, we could in fact increase their tolerance to the drug by about 60-fold. If we knocked out, in contrast, if we knocked out miR-147B with an antisense molecule, we could actually make those cells now more sensitive to osimertinib by about 166-fold. Some of that data is shown here, either in the sort of clonogenic assay or in the 3D culture assay, where you can see that the combination of these drugs leads to the fewest of either cells growing up or organoids growing up in these experiments. In addition, we showed in a mouse model that if we knock out miR-147B in cells grown in a xenograft, we can slow the growth of those cells in this mouse model. And the tumors are about half the size on average. So this suggested that miR-147B might be an oncogene in lung cancer, but also that targeting it might be useful in preventing tolerance in this particular setting. And so sort of to test that in a very preliminary way, we took a set of organoids in a similar way to what we had done in the pancreatic cancer case. These were organoids that we obtained from Carol Bolt at the Jackson Laboratories. And in this case, I'm just going to show you one particular patient-derived organoid from an EGFR mutant patient that had the activating mutation in EGFR plus the T790M mutation that made them resistant to alatinib, that's sensitive to osimertinib. And we asked, could delivery or pre-treatment with an anti-miR-147B, shown here by this alanine, it was a locked nucleic acid that we used in this particular case, could we see any potential benefit in pre-reducing the levels of miR-147B in these organoids? And so what you can show here is that untreated cells grown as organoids will just increase in size. Cells treated with just osimertinib obviously show a growth reduction because, of course, these cells are sensitive to osimertinib. But these organoids reduce in size to about half their normal size. But then over time, these organoids will start to grow back. What we found is that pretreatment with anti-miR-147B led to, first of all, a dramatic reduction in the number of cells in the organoids, suggesting that we were killing more of the cells with osimertinib. But we also found that, at least by the end of the experiment, we showed no regrowth of these tumors. So blocking miR-147B in this case prevents and delays drug tolerance in this particular setting. And we're hopeful that in the clinic, if this ever was to make its way into the clinic, that this particular pretreatment might be useful in preventing tolerance in patients that are treated with EGFR TKIs. So I didn't show you all of the data where we talked about which miR-147B targets were important in this particular setting. I did mention that miR-147B directly targets VHL, which of course is involved in the HIF-1 alpha response. We did go on to show that drugs that inhibit this particular or that inhibit or activate this particular pathway in defined ways can also influence drug tolerance. And so there's the potential now not only for anti-miR-147B to be used in an anti-tolerance strategy, but also for hypoxia targeting drugs to be used in this particular strategy. So I'm going to end with just some thoughts about how we're thinking about incorporating non-coding RNAs into standard of care. And I'm taking a risk here because obviously these are preliminary thoughts. There's a lot of barriers to doing the things that I'm proposing to do here, but I would love the feedback from the audience and your help in making this a reality if it's possible. So given what I've told you about from some of the small vignettes that I mentioned today, we do think that targeting microRNAs and other non-coding RNAs are going to be useful or potentially druggable targets in cancer and already are in clinical trials, and hopefully will make it into the armamentarium of oncologists in the not too distant future. But we believe that in order to make sense of or at least in order to sort of harness this opportunity, we need a complete microRNA-seq analysis to be formed on the tumor and also on the serum from each cancer patient. And we can do this currently at the non-coding RNA core. But of course, this could be done in any sequencing core. We need a complete DNA sequence of the tumor and the germline of cancer patients. This is the 98+2 part that I was talking to you about. And this is really where things become a little tricky. We really need an integrated bioinformatics and pathology analysis to quantify the malignant pathological phenotypes in the patient's cancer and to identify the significantly altered genomic and microRNA functions within cells, or non-coding RNA functions within cells. Then this pathology report that hopefully includes an interpretation of not only the pathology, but also the DNA and RNA data and the potential therapeutic vulnerabilities could be presented at tumor boards to aid in therapeutic decisions. Patient-derived avatars would need to be grown, and the therapeutic potential of different drugs that were suggested by this pathology report could be tested. In addition to traditional drug testing, we would obviously hope that we could also test anti-microRNA drugs in the same type of setting. And then the recommendation would be for possible microRNA-based therapy to be administered to the patient. Some of that could be done in a non-coding RNA core, which has a GMP facility, and we could package those antisense molecules into drugs and walk them from the core to the clinic. And then lastly, a searchable file of the DNAs of the patient's DNA, RNA, and prognostic data should be placed in the electronic medical record. I know that there are massive HIPAA problems with doing something like this, but I feel like there's probably an important part of the story, because we're discovering so much about non-coding RNAs every day that later reanalysis of this data could show some vulnerabilities that we didn't appreciate at this point, and this additional information could then be used at a later point in the treatment of the patient. So I'm going to end there. I hope that I've told you that we're on our way to making this a reality for patients. We still have a way to go, and I really value your help in this. And with that, I'm going to end by thanking the people in my lab who did the work. These are the current people in my lab. Everybody shown here in red, my former graduate students and postdocs, had a hand in some of the work that I told you about today. I'd obviously like to thank our collaborators, without which we wouldn't be able to do much of this work. And these funding sources for their help. Anyone shown in red has had a direct role in funding the work that I talked about today, including the SPOR from Yale, lung spore from Yale, the Dana Farber lung spore, the V Foundation, the Ludwig Center at Harvard, the HIRM, and the NCI. And with that, I'm going to end and take your questions. Thanks very much.
Thank you very much. That was really spectacular. And we're right at 1 o'clock, so I think we're going to have a very limited number of questions. I'll try to sort of combine a group. One, there are several questions around therapeutic index, and one I like particularly from Bob Weinberg is around asking if there are examples of microRNAs that target both an oncogene that seems to be essential, but also a tumor suppressor gene within the same cancer cell. And I guess to expand on that, the skeptic might say that microRNAs are by their very nature dirty drugs. So how do you respond to that?
Yeah, I think that is an excellent question. And in fact, one of the barriers that we recognized early on is that not only was there an opportunity here, but there was also an opportunity for both off-target and unwanted on-target effects, especially in cases where we know that a single microRNA can regulate multiple different, some of which are genes, some of which are tumor suppressors. So there are, in fact, examples like that. There are definitely examples where a microRNA can be oncogenic in one setting and tumor suppressive in another setting. One of the things that's really been important for the field to determine is what is happening in the individual cancer patients themselves. So not just in cell lines or in TCGA data, something like that, but what is actually happening in the patients? Can we determine whether those particular targets are in fact true targets in that particular cancer patient's setting? So in the case of MIR34, I would say the overwhelming number of targets in that case turn out to be oncogenic. But there are, some genes in that list that one would say might be tumor suppressive. But what is happening in the actual individual cancer patient's sample? So that's why it's important to do the entire set of microRNA sequencing and RNA sequencing from the same patient sample. So Bob brings up a really good point. These are dirty drugs. I mean, one way I've thought about this is instead of calling them dirty, rather calling them one-drug cocktails, because you can imagine that a single drug in this case could target hundreds of different things. The key is to find the microRNAs and the tumor type where those things that it's targeting are in the direction that you wish. But I think that we're getting there. It's not trivial, but we're definitely getting there. And certain microRNAs like 155, MIR34, there's an overwhelming evidence in the literature that these are going to be useful RNAs and act the way that we think that they're acting.
And so a great follow-up then to your last statement was, do we understand about mechanisms of resistance to microRNA-related therapies in tumors, and are there examples where the resistance mechanisms have validated the on-target activity of the microRNA therapeutic?
Right. Yeah, that's another excellent question. And in fact, it's part of a grant that I'm, that I currently have to try and work on microRNA-based resistance mechanisms. Some of the air for that study was sort of taken out when the trial for MIR34 was terminated. We were hoping that we would be able to look at patient samples, ones that didn't respond, and be able to ask whether there was, a pathway or a set of pathways that were being induced in some sort of resistance. I can tell you that right now there is very little literature on what are the potential mechanisms of resistance to microRNA-based therapies. It's something that we would love to work on more. And in fact, we have some of the models now where we could address that issue. I can tell you that in our small molecule screen for molecules that synergize with MIR34, we also find molecules that antagonize MIR34. And so we're currently looking at that in case they might provide some insights to what might be the mechanisms of resistance to this class. It is a very important point, and I think it's something that we're very interested in pursuing in the future.
All right. Well, at that point, I will thank Dr. Slack. We still have 500 attendees. I think we had nearly 1,000 at least again today. So for a very very enlightening lecture. Again, Dr. Ross Levine will be speaking tomorrow on clonal evolution in myeloid malignancies. And we hope everyone stays well until then when we see you tomorrow.
Thanks, everybody.